Photosystem 1 has P700. 700 is the wavelength absorbed by the reaction center, which is chlorophyll A. 700 wavelength is a deep red. Photosystem 1 is one set of reactions that best absorbs light at 700 nanometers. Photosystem 2 has a slightly different version of chlorophyll A in the reaction center. It has P680, which is more of a moderate red versus a deeper red. This requires light with wavelengths of 680 nanometers. When P700 absorbs light from the antenna complex, it becomes a strong electron donor or a reducing agent. The electron goes to an electron transport chain. When P700 loses its electron, it is oxidized. The electron circles back to P700 through a second electron transport chain. P700 is restored to its original state and is ready to absorb another photon. When P700 absorbs a photon and releases an electron, it needs to get an electron back before it can absorb another photon. This process is called cyclic photophosphorylation. The cycle of electrons creates a proton gradient across the thylakoid membrane that generates ATP through the process of chemiosmosis. This ATP is actually going to be used later in photosynthesis. It is not for the cell to use as energy elsewhere. That's the job of the mitochondria and cellular respiration. The protons move from the stroma, which is the space outside the thylakoids, into the intermembrane space of the thylakoid. The proton gradient provides energy to the ATP synthase, and ATP is produced. This ATP is going to be used later to make glucose. Photosystem 1 also has an alternative non-cyclic electron path which doesn't produce ATP. Instead, the electron from P700 is used to make NADPH. This is a derivative of NADH. NADH, again, is a reduced form of NAD. NADPH is an electron-carrying coenzyme that chloroplasts use to build glucose. The non-cyclic route provides most of the power to build glucose. When P700 gives up electrons to make NADPH, it needs another electron so it can absorb another proton. This is where photosystem 2 comes in. P680 in photosystem 2 absorbs light best at 680 nanometers. P680 acts as an electron donor when it is excited. The electron from P680 moves along the electron transport chain of photosystem 2 and oxidizes P700. Photosystem 2 also produces ATP when a proton gradient is created from its electron transport chain. This ATP production is called non-cyclic photophosphorylation to distinguish it from the cyclic type. But now, how is P680 oxidized? How does it get back on its electron? The electron comes from water. When photosystem 2 strips electrons from water, it creates the free oxygen we breathe. Until this photosystem 2 pathway evolved over 3 billion years ago, there wasn't oxygen in the atmosphere. By adding oxygen to the atmosphere, it caused the massive extinction of anaerobic organisms that only existed up until that point, but it opened up the door to allow the evolution of aerobic organisms. The light-dependent reactions of photosynthesis transform light energy into the chemical bonds of NADPH and ATP. The absorption of light splits water, produces ATP and NADPH. Oxygen is a waste product released through the stomata. ATP and NADPH remain in the stroma to be used later. To reduce each NADP to NADPH, 2 electrons are required. Photosystems 1 and 2 must each absorb 1 photon to provide 1 electron. So 4 photons must be absorbed to get 2 electrons. So the production of 1 molecule of NADPH requires 4 photons of light. As each electron goes from photosystem 2 to photosystem 1, enough protons are pumped to make 1 ATP. So 4 photons make 2 ATPs. The energy stored in the bonds of ATP and NADPH is used to make glucose. Melvin Calvin in 1945 at the University of Berkeley, along with his colleagues, worked out the set of reactions that converted the C atoms, the carbon atoms of carbon dioxide, into the carbon atoms of glucose. The Calvin cycle consists of 3 parts. The production of phosphoglycerate from the combining of CO2 with an acceptor molecule is the first phase, called carbon fixation. The second phase is the conversion of phosphoglycerate into glycerol aldehyde phosphate in 2 reactions that use ATP and NADPH. Those, again, were produced in the light reactions that we just went over. The second step is considered reduction, and the third step is the regeneration of the initial CO2 acceptor, which is RUBP. If we look at it in a little bit more detail in part 1, which is carbon fixation, carbon dioxide combines with ribulose biphosphate, which is a 5-carbon, and it forms an unstable 6-carbon compound, which immediately breaks down into 2 PGAs, or phosphoglycerates. Those are each 3 carbons. The enzyme Robusco that catalyzes this reaction works slowly, so plants tend to make a lot of it. It makes up about 50% of the total proteins in the chloroplast. Robusco is the most abundant protein on the planet. In part 2, phosphoglycerate, which is PGA, is converted into glycerol aldehyde phosphate, which is G3P. That's a 3-carbon sugar. You can consider G3P as half of a glucose. Part 2, which is called reduction, requires 1 ATP and 1 NADPH for each PGA. Each CO2 produces 2 PGAs, so 2 ATPs and 2 NADPHs are used. Some of the G3P enters the cytoplasm and are converted to glucose. So when we create 5 G3Ps during reduction, 1 of them leaves and the other 5 remain. In part 3, the regeneration of RUBP is the messy part of the Calvin cycle. It really requires 7 different enzymatic steps, which aren't shown here, but the result is taking the 5 G3Ps that are left and converting them back into 3 RUBPs to start the cycle again. For each molecule of glucose that's produced, 6 CO2s must enter the Calvin cycle. As the cycle turns 6 times, it produces 12 G3Ps, 2 of these form glucose, and the other 10 regenerate into RUBPs. Each turn of the Calvin cycle uses 1 RUBP times 6, 2 ATPs times 6, and 2 NADPHs times 6 to create 1 glucose. It also costs 6 ATPs to make 6 RUBPs. Therefore, 1 glucose uses 18 ATPs and 12 NADPHs in order to make that 1 glucose. And again, the ATPs and NADPHs were formed during the light reactions. The Calvin cycle is often called the dark reactions because light is not required. There are many factors that influence the productivity of photosynthesis. One of them is wavelengths of light. Light that can't be absorbed can't be used. So the light that is outside of the wavelengths that the different pigment molecules can absorb cannot be used. The light intensity, okay, this can involve things like cloud cover and shade. Plants don't make as much glucose in the shade. The length of the day. The length of daylight is much greater in the summer than it is in the winter. So plants are usually much more active in the spring and the summer. The length of the growing season. Depending on where you live in the world, the closer you get to the equator, the longer the growing season or the longer your spring and summer season is. The angle of the sun, as you know, changes with the different seasons, and it's most direct in the summer. Environmental conditions like fog, which filters sunlight, other high amounts of pollution in the air, or even layers of vegetation like we find in a rainforest can affect how much photosynthesis occurs. The availability of water can influence photosynthesis. Water carries nutrients and it evaporates from the leaves to regulate the plant's temperature. When it's hot and dry, the stomata opens in the leaves and closes to conserve H2O, but CO2 and oxygen can't enter and leave when the stomata is closed, trying to conserve water. Desert plants need to keep the stomata closed a lot, so getting enough CO2 for photosynthesis is a challenge. Low CO2 levels can cause photorespiration, where plants actually waste glucose. What happens is the plants use ribosco, that's the enzyme used when CO2 enters the Calvin cycle, to convert RUBP into 2 PGAs, but when CO2 is low, plants convert RUBP into 1 PGA and one other molecule called phosphoglycolate. The plant winds up wasting half of the glucose it produces. Some plants have evolved to maintain high levels of CO2 inside their cells when their stomatas are closed. They have special enzymes that do this. In these plants, the carbon dioxide doesn't produce PGAs, but it produces a 4-carbon oxaloacetate, same as what we saw in the Krebs cycle, which enters a special cell. It breaks down and releases CO2, which can then enter the Calvin cycle. So if the plants do the normal Calvin cycle, they're considered C3 plants because they make PGAs, which are 3 carbons. The plants that actually make the 4-carbon oxaloacetate and then produce CO2 from it are called C4 plants because oxaloacetate is a 4-carbon molecule. Corn and sugar cane are examples of C4 plants. And then there's CAN plants. Here we have a pineapple. Cactus is another example. It uses a modified C4 pathway. What they do is they open their stomata at night when the air is cool, water loss is minimal then, and they store the CO2 from the evening until daytime when sunlight is available for photosynthesis.